although fungal cell walls are made out of chitin instead of cellulose. Now, the animal cell has some features that you don't typically find in plant cells. The flagellum is rarely found in plant cells. Why? Well, wait a second. The flagellum is used to make the cell mobile, to move it. So plant cells do not really move around very much through a liquid medium, although some plants do have flagellated sperm. So, the animal cell also has a nucleus. It also has a nuclear envelope which surrounds the nucleus. It has an endoplasmic reticulum, so a rough and smooth. Technically speaking, the endoplasmic reticulum is not nearly as small as you see here. The endoplasmic reticulum actually extends throughout the cell. It's just they draw it relatively small so that it's really obvious what's what. For example, the Golgi vesicles, or Golgi apparatus, is a stack of flattened vesicles. It is not that hard to manage to mix it up with the endoplasmic reticulum if you are not careful. Hence why they draw it in different colors here. Some ribosomes float around freely. Why is this the case? Well, that's because, wait a second. If we know that the cytoplasm is full of proteins, where did those proteins come from? Well, they're manufactured on these free ribosomes. It's the proteins that get exported or need to become membrane proteins that end up attaching to the rough ER. So, peroxisome. Now, peroxides are harmful to cells. The great oxygen catastrophe, or the great oxygenation event, depending on how you want to call it, actually wiped out most of the life on Earth, because most of the life on Earth was anaerobic and not adapted to oxygen, and oxygen destroyed their organic molecules. Fortunately, life, as we know it now, is adapted to oxygen. However, we do point out that the great oxygenation event was a mass extinction. So, the peroxisome is, for the most part, intended to deal with peroxides and other harmful radicals. So we do point out that in plants, the peroxisome is known to contain a crystal of catalase. Catalase was one of the earlier enzymes to be discovered. It is used to break down hydrogen peroxide. Now, your cells also contain catalase. As you know, when you add hydrogen peroxide to an injury and it bubbles vigorously, that is catalase doing its work, breaking it down to water and oxygen gas. Now, here we note that there are actin filaments, there are microtubules, and there are also going to be microfilaments inside an animal cell. But for now, we don't really need to be concerned about the cytoskeleton for grade 10 purposes. Alright, here we list the components that we really need to know for grade 10. So we have the cytoplasm, the solution inside the cell membrane. So it's water and a lot of proteins and some dissolved carbohydrates and some lipids as well. Hence why steroid hormones can cross through the cell membrane and then go straight into the nucleus. They don't teleport. They still need to float through this solution. Now, it behaves as a sole gel in that it has parts that behave more like a liquid. That's why cytosol is often used as a substitute term for cytoplasm. And parts that act like a gel. The cell membrane is the membrane that surrounds the entire cell. Sounds really obvious, right? Yeah, well, the thing is, the cell membrane, depending on the particular cell's orientation relative to other cells, it could be uniform, such as, say, for an amoeba, where it's a unicellular organism. Or for, say, an intestinal lining cell, it has an apical side and a basolateral side. The composition of phospholipids for the apical side and the basolateral side are different. which help it satisfy various different functions. Now, the cell membrane is capable of pinching inward in phagocytosis or pinocytosis. Phagocytosis is eating, so to speak, like gobbling up another cell or a large cell fragment. And pinocytosis is taking a drink, so to speak, of a droplet of extracellular fluid. The extracellular fluid could be just water with some dissolved ions, or it could be, as is the case in, say, blood plasma, it could be filled with all sorts of proteins. Relatively. Of course, water is still a major component. And then there's a nucleus. Here are some cells. They have been stained for their nuclei, and this cell here is dividing. The nuclear membrane has dispersed. The chromosomes have condensed. Whereas here, these two cells are not dividing. They are in interphase. Now, eukaryotic cells do not always have their nuclei, as is the case in this one. During division, the nucleus is dismantled temporarily. It reassembles afterward into two separate nuclei. Now, DNA, Deoxyribonucleic acid, is a material in the nucleus that contains genetic information as we know it. It is possible that early in life's history there was an RNA world. Instead of the deoxyribonucleic acid, we used ribonucleic acid instead. And, well, the thing with ribonucleic acid is that, yes, okay, you can still have double-stranded RNA. In fact, many viruses operate off of double-stranded RNA. But currently, we use DNA because it's more stable. Thus, it can keep information better. It mutates less. And now, when we encounter double-stranded RNA, our cells chop it up because they think it's a virus. Therefore, chop, 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 chop. Try to kill it. Even though technically viruses are not alive. So mitochondria is the powerhouse of the cell, and it is the site at which the oxidation of pyruvate to carbon dioxide and water is completed. Now, mitochondria, yes, okay, we draw it mostly as we did above, but on electron micrographs, you might find that the pockets inside are a lot denser than you might initially think based on, well, what we drew up there. So I won't draw the rest, but I hope you get the point. So how do we distinguish this from the Golgi apparatus? It's because the Golgi apparatus typically flares out a little bit on either side, and it's the flattest in the middle. Whereas this, well, there's still going to be a most often cylindrical shape. Now, mitochondria most likely arose by endosymbiosis of alpha proteobacteria. So these were bacteria that could use oxygen to make energy, and they got eaten up by an early eukaryotic cell that couldn't use oxygen to make energy, but it could tolerate oxygen, hence why it's found in the same environment. Now, it ate it up, and it found that cooperation was useful. So, what about chloroplasts? Well, chloroplasts also have a small ring of their own DNA, like mitochondria do. Chloroplasts are another event where the early eukaryote, now is mitochondria, ate, most likely, a cyanobacterial cell. Now, most eukaryotes did not eat a cyanobacterial cell, so they do not continue down the lineage to photosynthesizing. We do note that there were secondary endosymbiosis events as well. For example, some plastids in some lineages, i.e. photosynthetic organelles, plastids, some of them have not two membranes as you would expect for, oh, I eat a cell. The membrane I put around it when I was eating it, and the membrane it originally had, well, those are two membranes. Some plastids don't have two membranes. Some plastids have three. That's because they started off with, like others, that have four, even today. That's because, well, wait a second. If this gets eaten up by another cell, well, let's just abbreviate the other cell for scale, and that gets eaten up by another cell, what happens? 1, 2, 3, 4. Of course, it didn't happen to mitochondria, but in the case of certain algae, so-called algae, even though technically they're not usually not algae, well, this did occur. Now, the endoplasmic reticulum. The endoplasmic reticulum, also known as ER, comes in rough and smooth varieties. The smooth variety typically appears as a bunch of tubules. It is where lipid assembly takes place in the cell. The rough variety comes usually as a bunch of flattened sheets. Now, the edges of the rough endoplasmic reticulum regularly pinch off and go to the Golgi apparatus, where they merge with the Golgi apparatus, empty out their contents, where these contents, the proteins that are to be secreted, are further processed. And in some cases, they're not to be secreted. They're actually embedded on the membrane. They are processed further. They move up the stack from the side closer to the nucleus to the side further out. And eventually, they get exported to the surface of the cell, where they may be released into solution, or they might stick to the surface. Now, the rough ER has pores in it. These pores are where the signal recognition particles that attach to ribosomes that have a certain piece of polypeptide hanging out as a signal. And these SRPs will attach the ribosomes to these pores and make sure that they translate into these pores instead of just translating the protein into the cytoplasm. The Golgi apparatus is a flattened stack of sacs. Each sac has certain enzymes, proteins, that conduct particular modifications to whatever you put in there, or rather whatever is exposed into there. So what happens to the vesicles is that they come in one end, they attach, they eventually butt off again, they go to the next one, they butt off again, and so on, until they reach the last one, and then they go off to secretion. This is an electron micrograph, given the 30,000 times magnification. And this is false color to make the Golgi more visible. Now, vacuoles. Vacuoles do not, like the nucleus, it does not, also does not, require an electron microscope to see. Technically, you don't need an electron microscope to see chloroplasts either. Why? It's because the little green things that are moving around inside a cell under the optical microscope, they are pretty obvious, right? So the nucleus is really visible, but the vacuoles are also really visible. There may be more than one. Now, vacuoles, smaller vacuoles are called vesicles. Technically speaking, they're all endosomes, but for now we can know them as vacuoles and vesicles. They vary greatly in terms of function and size. Peroxisomes can be considered a type of vesicle. Lysosomes, which are used to digest stuff inside animal cells, they are by definition not existent inside plant cells. Even though there are endosomes, vesicles, that are used for the exact same thing in plant cells, by definition they are not called lysosomes. I know that this sounds incredibly stupid. So, lysosomes are used in animal cells to merge with the vacuoles that have just been formed by engulfing, say, a bacteria or something else that you want to digest. And what else do lysosomes do? Well, if you make a membrane around an old damaged organelle, old or damaged organelle that is no longer functioning properly, you make a membrane around it, the lysosome merges with that membrane, and the old or malfunctioning organelle is degraded. It is digested and the material can be recycled. Now, plant cells have a cell wall, which animal cells do not. essentially, for the most part. We do note that fungal cells also have plant cell walls, but they're made out of chitin, which is, by the way, the same material that insect exoskeletons are made out of. Chitin is used in surgical threads that you want your body to be able to digest away after it has served its purpose. Now, plant cell walls are cellulose-based. Cellulose is made out of a lot of glucose linked end to end to end to end to end, and so on. Starch is also a lot of glucose linked end to end. So why is cellulose indigestible to most animals? Well, all animals technically, because we need to have bacteria or fungi that can digest cellulose to actually digest it. So why is it indigestible to animals? It's because the orientation of the bond between the glucose molecules is different from starch. So animals can't digest it. The result is that cellulose is a very durable material, and it can persist long after the plant cell has died. Now, you probably have quite a bit of cellulose at home in the form of slabs of processed dead tree, also known as books. The large central vacuole, or usually central, and there may be more than one. The large central vacuole is fairly characteristic of plants. Animals, for the most part, do not have this. Although we do note that certain floating organisms, certain types of plankton, do have oil droplets that helps them regulate their density so that they don't sink to the bottom too fast. Plants also have chloroplasts, which are very, very obvious even under a relatively low magnification on an optical microscope. These are what give plant cells their green color. For the most part, plant cell walls tend to be, well, anywhere from colorless to, okay, there are a few cases where they are actually greenish, to brown. For example, ebony. Have you seen ebony wood? There's a reason ebony is considered a shade of black. It's because it's just so dark in color. Mahogany is not particularly light either. Now, in summary, the cell theory states that all living things are made of cells. So cells are the basic building unit of life. The cell is the simplest unit that can carry out all life processes. And all cells are reproduced from other cells, at least at present. In the distant past, perhaps there was a time when abiogenesis created what can be considered a protocell from non-living matter. But that took a very, very long time and a very, very large number of attempts and accidental reactions for it to possibly happen. Now, the simplest single-celled organisms, including bacteria, are prokaryotes. Calling them simple, though, is a little bit of a misnomer, given that the life in general is actually quite complicated. So, comparatively simple organisms is probably a better term. More complex organisms, including multicellular ones, are eukaryotes. Now, there are some multicellular prokaryotic forms, but for the most part, prokaryotes are single-celled organisms. And there are plenty of eukaryotes that are single-celled as well, for example, amoebas, or paramecium. Eukaryotic cells contain organelles that carry out specific life functions. They're called organelles because, well, they wanted to name them little organs. The cell membrane, cytoplasm, nucleus, mitochondria, the smooth and rough endoplasmic reticulum, the Golgi apparatus, and the vacuoles occur in both plant and animal cells. The cytoskeleton also occurs in both plant and animal cells. But in plant cells, we can find a central large vacuole, usually, We can find cell wall, usually. And we can find chloroplasts, also, usually. There are plants that have, well, any combination of lack of these features. Animal cells have flagella. Plant cells, for the most part, will not have flagella. Or, well, actually, animal cells, for the most part, don't have them either. But plant cells, for the most part, cannot have flagella. There are a few plant species with flagellated sperm, and that's it. Whereas many different types of animal cells and protists have flagella. So, we do note that the flagella you find in eukaryotes and the flagella you see in prokaryotes are very different things. In prokaryotes, a flagella is basically a long string of the protein flagellin with a rotor at the end. Now, in eukaryotes, it's a bundle of specially arranged microtubules that flex and side to side. So, a prokaryote spins around using its rotor, whereas a eukaryote will lash side to side. This is a very important distinction. So that's it for now for the basics of plant and animal cells, and we'll see each other again next section.